everybody. Oh, hello. Okay, great. Whoops. So I would like to talk to you about a multivariate approach to the imaging genetics problem that you've seen a number of times. And we try to create a morphological brain atlas of multivariate genetic depression risk. The first thing that you have to know is that recently there was a large genome-wide association study conducted in 130,000 depressive people and 330,000 healthy controls. And I mean, what was found there, it's just been published a couple of weeks ago in Nature Genetics. What was found there is that we identified 44 gene loci with genome-wide with significance on the genome-wide level. So the interesting thing is that all of the gene expression and everything that you see here, there, that only brain tissue showed significant enrichment. So the interesting question is, can we get more detail on that? Can we figure out what the connection between the 44 risk genes that were identified in this large-scale analysis and brain morphology is? And what we did then is we first used the classic the univariate imaging genetics approach, which just takes the 44 genetic loci and the 157 free surface parameters that we used in this analysis. It's volume, surface, thickness, just the standard free surface parameters that you probably all know, and try to correlate those in 1,184 depressive patients. And we found it a surprising thing that even given this large data set, and I think it's one of the largest MDT data sets that has been tried in the imaging genetics literature, we didn't find anything. We do a correction, and we did a number of different types of correction, and so on. The picture is very clear that there is no association between a single genetic variant and any of the 157 parameters, which surprised us, especially given my last slide that clearly showed that we should find something in the brain. So we thought, okay, actually, if you think about it, the hypothesis that almost every researcher would have is that it's not a single gene or not a single genetic variant that would affect depression or that would affect the brain in the end, but that is actually the multivariate pattern of multiple genes. So we took the 44 genetic variants and tried a machine learning approach, a random forest to be more specific, in a repeated cross-validation framework, and tried to predict from the 44 genetic variants, from the pattern of the 44 genetic variants, each single one of the 157 free surface parameters. And if you do that, it's probably a little small back there, but if you do that, the blue histogram shows you the random. We conducted a permutation test with 157,000 permutations, and this is the histogram of all the random ones. And what you see in red is those areas that still showed a significant association. So now we all of a sudden find a significant association between the patterns of the 44 genetic loci and the number of the 157 free surface parameters. So the next thing that we looked at then was, well, what if you check how many times a certain genetic variant is relevant in the predictive pattern of one of the 157 free surface parameters. While you would expect basically with 44 genes and 157 parameters, you would expect something between three and 4 occurrences. You see that there are some that had a frequency of higher than 40, and quite a few had an occurrences higher than 20. So that made us think that kind of sparked the question, if there is a similarity in the predictive genetic patterns across the parameters in free surface. And we checked this by first checking whether we can summarize the predictive patterns in some way. If you look at the variance across the 157 free surfer parameters, we checked if they're similar in some way. And what we did in this case is we conducted latent semantic analysis, which I will just, in the interest of time, just skip over a little bit. But what is basically done is you do an SVD, a singular value decomposition, and what you arrive at is basically a lower dimensional representation of the predictive patterns. And then we checked what would be a good representation, and as you can see here, it's the number of components in the SVD, and you can see here that 3 seems to be a reasonable number. We also did this in 2, and we did this in 4, but as this graph is showing, 3 is probably a reasonable number for this number of components. The results don't change relatively little if you take 2 or 3 or 4. So we took these 3 components and asked the question, are brain morphological parameters, the 157 that you get from FreeSurfer, are they similar with regard to the latent genetic factors that we just found in that latent in the semantic space? So if you do that, we're surprised to see how well this works. That even with the naked eye, you can easily see 4 distinct clusters. And what we used, of course, here is dbScan, density-based spatial clustering applications with noise. It's just a standard clustering algorithm, and that came up with this clustering. But even if you didn't have the colors, you would instantly see that there is at least 3, and probably 4 if you rotate it a little bit, is 4 distinct clusters. This means that looking at the predictive patterns, we can basically group the brain morphological parameters from FreeSurfer into 4 different groups, where each of the single groups of the single clusters here has a highly similar pattern, has a highly similar predictive pattern of genetic variants that drive the prediction of this area in a multivariate sense. So now, of course, it's nice if you have a clustering, and so far it's all a data-driven approach, and could convince you, or it just couldn't. But we were looking for something that would externally validate these clusters. And what we did in our final analysis then is we used the Neurosynth platform, which most of you have probably heard of. If not, check it out on the web, it's great. What this does is you can give it a mask. We use the decoding feature. You can give it a mask of an area, or you can give it a brain map, and it will give you the association of more than 3,000 terms with this map. And that's what we did for each one of the FreeSurfer areas, for each one of the morphological brain areas. And then we computed the median term association for each term, for each term across the regions in one cluster. So we take the median of the term associations in cluster 1, the median in cluster 2, the median in cluster 3. And then we get the median term association, and then we repeat this in a permutation test 10,000 times with random clusters. So we just drew random clusters of the same size. And then we checked and saw that for all 4 clusters, we find a significant semantic term consistency, meaning that our term consistencies in each one of the clusters are well above chance, are well above chance in terms of if you just drew clusters randomly, you would not see such high term associations. And keep in mind that Neurosynth is an independent platform, and all that happened in this analysis is we used the Neurosynth platform and got the term associations with the FreeSurfer Atlas, and that's it. And still it maps onto our clusters. And so we think that we might be onto something here. Of course, the analysis is not finished by far, but still we think that there is a few important messages here. We think the first thing to see is that we could predict from the pattern of the 44 risk genes that had previously been identified in a large-scale study, we could predict the brain morphology. The univariate analysis, the classic imaging genetics analysis, did not find anything, even though this is one of the largest samples ever used, where you would expect that this power would actually help you to find something. And brain morphology is organized into 4 distinct clusters, each with a similar predictive pattern. And the 4 clusters carry above chance semantic meaning. And with that, I'm just finishing in time. Thanks for your attention.